In a healthy nervous system, signals from the brain are transmitted to the spinal cord by upper motor neurons, and then to the skeletal muscles through lower motor neurons. Spinal muscular atrophy, or SMA, is a rare genetic disease that originates in the central nervous system in lower motor neurons. Degeneration of these neurons results in muscle atrophy that can impact motor, respiratory, and bulbar functions. As the disease progresses, this can restrict basic abilities like moving, breathing, and eating independently. For the most severely affected patients, symptoms become apparent during infancy. These patients are unable to reach motor milestones like sitting independently, and typically do not survive beyond 2 years of age without respiratory support and nutritional interventions. Patients with less severe forms of SMA may first experience symptoms as infants, children, or adults, and are more heterogeneous in their ability to reach key motor milestones. Some patients may only achieve independent sitting, while some may walk independently. However, since SMA is an inevitably progressive disease, over time patients lose functional abilities they had once gained. SMA is traditionally classified according to 4 types based on age at symptom onset and motor milestones achieved. While symptoms of SMA may manifest at different times for individual patients, there is ongoing progression of subclinical disease activity that occurs prior to symptom onset. Symptoms become apparent only after accumulated damage to motor neurons has caused a critical loss of functional motor units. The cell bodies of lower motor neurons are located in the anterior horn of the spinal cord. These neurons are dependent on a protein called survival motor neuron, or SMN. Lack of SMN protein underlies the fundamental deficit in SMA and leads to loss of motor neurons and muscle atrophy. SMN is a key protein that functions in a wide range of important cellular processes, including RNA metabolism, DNA recombination and repair, signal transduction, intracellular trafficking, endocytosis, and autophagy. There are 2 genes that produce SMN protein, SMN1 and SMN2. SMN1 produces fully functional SMN protein, while SMN2 produces only a small amount of fully functional SMN protein, accompanied by mostly non-functional SMN protein, known as SMN delta 7, that is rapidly degraded. In SMA, homozygous disruption of the SMN1 gene leads to a significant loss of functional SMN protein, as mostly SMN delta 7 is produced in the body from the SMN2 gene. This is not enough to maintain the survival of motor neurons, which leads to the development of SMA. The number of SMN2 genes naturally varies from person to person due to gene duplication or conversion events. SMA patients possess between 1 and 6 copies of SMN2. The degree of severity of SMA generally correlates with the number of SMN2 copies a person has and amount of SMN protein they produce. The SMN1 and SMN2 genes are nearly identical, with only 5 nucleotide differences between their sequences, none of which alter the encoded sequence of amino acids. However, one of the nucleotide transitions disrupts splicing, a process that removes non-coding introns from the pre-mRNA transcript. Incorrect splicing results in exon 7 being removed, along with the introns. Exon 7 exclusion affects approximately 80 to 90% of mRNA from SMN2, meaning only 10 to 20% of the protein made is functional. SMN is normally expressed throughout the body, but the role of SMN protein in non-CNS tissues is not fully understood. As the fundamental deficit in SMA, SMN protein production has become a key focus for therapeutic strategies that aim to alter the disease trajectories of SMA patients across the disease spectrum.